Great. Well, welcome everybody, and thank you so much for joining us this evening. We're really excited to be kicking off the 2024 season of the Bumblebee Atlas, and excited to share with you all our next phase that we're stepping into and how you can be involved in it. Tonight is a session for people who've been involved in the Bumblebee Atlas in the past, and also for people who've never heard of it before. So either way, wherever you are in that spectrum, welcome, and we're super excited to have you here. A few quick housekeeping things. This is a 2-hour webinar, and we're planning to get through the content with some time for Q&A at the end. So if you have questions, feel free to drop them in the Q&A category, which if you go over, I believe it's on the top of my screen. It may be on the top or bottom of yours, but there's a box that says Q&A. So feel free to put any questions you have in there. And then there's also a chat function. So if you want to write things that other people can see, go ahead and write those in the chat. There's also closed captioning available. So on that same bar, you should be able to turn on closed captioning if you'd like to use that. We're also recording this webinar. So if you need to step out early or you want to revisit content or share it with someone else, that recording will be going out after the webinar. For those of you who don't know, the Bumblebee Atlas is a program or a project of the Xerces Society for Invertebrate Conservation. And Xerces is an outfit that was founded more than 40 years ago and named for the now extinct Xerces blue butterfly. And we have a mission to protect wildlife through the conservation of invertebrates and their habitats. We have a whole range of really fantastic staff working on different projects across the country. And a few of those kind of projects and program areas involve native pollinators, endangered species, aquatic invertebrates, butterfly conservation, and also pesticide reduction. We are a member-supported organization, which means that the work we do depends on having wonderful donors who donate to help us support our organization. So we want to say thank you so much to people who do donate, and if you are a member, thank you. If you aren't, we hope that you'll consider becoming a member by going to xerces.org/donate. You can learn more about how to do that. Xerces is also fortunate to receive support from a wide range of agencies, organizations, businesses, foundations, and individuals. So I just wanted to throw up this list. I always find it heartening to see how many supporters we have, and give a big thank you to everyone who makes this important work possible. So before we jump into today's presentation, I just want to introduce myself and Rich Hatfield, my co-presenter. My name is Molly Martin, and I'm an endangered species conservation biologist with Xerces. I'm based in Portland, and I'm involved in a range of different projects, mainly focused on bumblebees and butterfly conservation. And as the Atlas transitions into this new phase of long-term monitoring, which we'll be talking about today, kind of what that transition means, I am going to be co-leading the project with Rich, and Rich he'll introduce himself, but he's been running the project for the last 6 years. So I'm stepping into co-leading it with him. So I'll hand it off to you, Rich.
Super. Thanks, Molly. Yeah, nice to see some familiar names and some unfamiliar names as well in the participant list. So thanks for joining us. My name is Rich Hatfield. I'm a senior conservation biologist. I'm also based in Portland, but I lead our bumblebee conservation programs nationwide. So I'm now coordinating Bumblebee Atlas projects in 20 states, which has reduced my ability to be active directly on the ground here in the Pacific Northwest in Oregon, Washington, and Idaho. So, Molly's jumped in to help me take on this role of implementing this new program, which we're excited to share with you tonight. And I'm gonna jump in. I think I can start sharing my screen over you here, Molly. And then if I could just get an okay from you or a thumbs up that you can see my screen, that would be great. Sorry, I missed it. Did you give me a thumbs up? Yeah, okay. Super. So before we look forward, we're just going to briefly look back on all that's been accomplished over the last few, over the last 6 years. We launched the Pacific Northwest Bumblebee Atlas program, as many of you know, back in 2018. It was a collaboration between the Idaho Department of Fish and Game, the Washington Department of Fish and Wildlife, the Oregon Department of Fish and Wildlife, and the Xerces Society. It was funded by a competitive state wildlife grant through the Fish and Wildlife Service, and also supported by the U.S. Forest Service. I'm sorry, if I said U.S. Forest Service, I meant to say U.S. Fish and Wildlife Service. It was also supported by the U.S. Forest Service and the Bureau of Land Management, and then a bunch of the supporters on the list that Molly showed a couple of minutes ago. The program ran for 6 years, and we engaged more than 1,600 participants who gathered close to 50,000 bumblebee observations and truly transformed our understanding of the status of bumblebees in the Pacific Northwest. And we're now at a space, the original goal of that program was to establish a baseline of information so that we can move forward and start enacting evidence-based, on-the-ground conservation actions. And we've begun to do that. Molly has helped spearhead the development of a bumblebee conservation strategy for the state of Washington. She's in the process of developing a pollinator conservation strategy for the state of Oregon. And we're generally using these data in many, many effective ways. This is a shot of a dashboard from our new website where we've shared all of the data from the Bumblebee Atlas project, where you, the volunteers and the general public, can interact with it, but also where land managers can interact with it and learn about different habitats for different species of bumblebees, learn about the plants that they're using, and see what species you can find in your region. So I would encourage you to go to our new Bumblebee Atlas website and check out some of the resources that are there. But as you can see here, you know, over the course of those 6 years, we had 1,600 people participate. We gathered more than 44,000 bumblebee observations, and over 3,500 surveys were conducted in the course of that work. And if you look at the spatial distribution of those records, you can see that we've really covered the entire 3-state area and have a really great understanding of the baseline distribution of bumblebees. The next phase for this program is for us to say, okay, we sort of understand how bumblebees are doing. We have baseline information about them. And now we need to step into more of a long-term monitoring protocol where we can identify species of conservation concern, which we've done, and then keep tabs on them. So we sort of know where they are, and we want to be able to know if our conservation efforts and or threats are changing their population status. So what we've decided to do is we've been working with some data scientists to look for some high quality monitoring sites across the 3-state region. We've selected 75 sites across Oregon, Washington, and Idaho, 25 in each state. And the goal will be to have volunteers go out and conduct the same Atlas surveys that we've done in the past. I say the same, there will be some slight tweaks, and we'll get into those later tonight. But to go out and conduct repeated surveys in those smaller grid cells. So where the goal is to gather at least 4 surveys in each sentinel monitoring area over the course of the year. And from that, we'll be able to learn a whole lot about individual species detection. And then from that, be able to build a better, sort of more informed long-term monitoring effort across the state, where we'll be able to say exactly when people should go look for X, Y, or Z species, and then have additional sites where people can visit in future years so that we can keep tabs on these animals and practice what's called adaptive management if we need to. So if we start to see changes in population status, we can actually know that more or less in real time and start acting to try to reverse or improve trends. And so that's the goal. of where we're going. This year in 2024, our two focal species are the western bumblebee, Bombus occidentalis, and Morrison's bumblebee, Bombus morrisoni. These are sort of the two imperiled species that are most common in our region, and so we're able to know where they live on the landscape from the efforts that you all put in over the last 6 years, and then go out to those sites where they were found and look at how they're persevering in those sites and learn more about their habitat and potential threats there on the landscape. So the goal again is to set aside these 75 monitoring sites, to have folks adopt them, and then to head out into those monitoring sites 4 times, approximately every 3 weeks between June 1st and the end of August. And we'll get into more of those details later. This is just a brief overview of what the program is going to look like in the years ahead. So again, the objectives of this program are to gain a better understanding of species-specific detection probability. That sounds fancy, but basically all it means is we want to learn how much effort it actually takes to find these animals at known occupied sites. So if we know that we're there and we go back there 4 times, we'll learn how long our surveys need to be, what the best time of year needs to be, so that we can keep tabs on them across a broader area across the state, and learn more, if we don't detect them, how likely it is that they could or could not be there. We also want to better understand sort of the landscape scale threats for these animals. And so going back to the same exact site repeatedly throughout time, within a year and between years, will allow us to also track management actions and threats at a landscape scale that could help us understand what is driving the population trends that we're seeing at those individual sites. And so there is the ultimate goal at the end there, the third bullet point of, we want to be able to track this occupancy over time. And this will let us see whether individual species are increasing or decreasing in population numbers, and allow us to work on those species if we need to. And I just want to, most of you probably know this, I said this in my introduction, but these efforts that we've spurred here in the Pacific Northwest have blossomed an incredible program that has spread across the country. We're now active in 2024 in 20 states. We have multiple partners and a number of funders that are interested in this work, and the work is going to inform on-the-ground cancer conservation for bumblebees at a continental scale. And it is truly inspiring and driven by all of the hard work from the volunteers that we've been able to engage in this work. So it's been a wonderful partnership. I think we're getting a lot out of it. And from what we're hearing from our volunteers, you all get a lot out of the program as well. So from before we even get started, thank you for being here, and thank you for participating in the Pacific Northwest Bumblebee Atlas. It's really created a wonderful movement that I believe will continue well into the future. And Molly and I get to work with this incredible team of people. So we have people spread all throughout the country that are running these Bumblebee Atlas projects, and we get to work alongside each other and take sort of what we created in the Pacific Northwest and make it better. And so all of these people are improving upon what we have done in the Pacific Northwest and gathering data. And likewise, with this long-term monitoring protocol, we're spearheading that here in the Pacific Northwest as well. So this will be the first time that this kind of landscape scale program has been implemented. And so we'll learn a whole lot more about what makes it work and how well it works, and how well the data provide for us the information that we're trying to get out of the back end. So thank you also for helping us to be the guinea pigs and learn how to implement this program. And, along the way, please reach out to Molly and I and let us know, what resources that you need. Or if we're not giving you what you need to participate in this program, let us know so that we can improve upon it and learn from those mistakes that we're making. So that's sort of a just real brief back look at the Pacific Northwest Bumblebee Atlas and a brief look of where we're going forward. The outline for today is, again, assuming you've never participated in the project, we're going to walk you through more or less how to conduct a survey from sort of A to Z. The goal is to sort of get you started from what it takes to adopt a grid cell, and then go all the way through conducting a survey and then submitting your data online. And even if it feels like you've done all these things before, there are some tweaks in our protocols for the long-term monitoring protocol that we hope you'll stick around to hear. And then in case you don't know, we've launched a completely brand new version of Bumblebee Watch. And so the data submission process is also a little bit different, and will be worth sticking around for. So thanks for being here. The outline you can see here, we're gonna talk about Bumblebee Watch, how to adopt a survey site, and then how to conduct a survey, including planning, collecting the Bumblebee data, collecting the habitat data, and then submitting your data online. And I think with that, normally in a lot of these presentations, we, will often talk about bumblebee ecology and conservation and identification. And we've sort of started doing that at a landscape scale or a national scale. So we do this ecology and conservation webinar. We taught one last month in April. It's up on the website it's on the Bumblebee Atlas website, and it's also on the Xerces Society YouTube channel. So if you want to learn more about bumblebee ecology and conservation and why we're doing this work and why we're directing our survey in the time period that we are, I would encourage you to go check out those resources and watch those webinars. And on the Xerces YouTube channel, there are just at this point a whole host of other bumblebee and frankly a lot of other invertebrate conservation stories and videos to watch. So would encourage you to check out those resources. There's also a lot of identification workshops that are up on there. Leif Richardson and I have taught how to identify bumblebees in the Western US for the last 2 or 3 years running, and we just did one on males last fall. So would encourage you, if you want to dive deeper into ecology, conservation, and identification, to seek out those external resources. And I think with that, I'm going to pass it back to Molly, who's going to start you on this journey of ours tonight.
Sounds great. Thanks, Rich. I'm going to try to take over screen sharing quickly.
And Molly, I see there's a question here. I haven't looked at it yet, but is it something? Okay. Robin, we'll follow up with you one-on-one about your question. That's a good one.
Are you seeing my screen full size?
It looks good to me, Molly. Yeah.
Okay. Sounds good. Second, I'm losing my mouse between all of my different windows. Great. So now that Rich provided an overview of kind of the Atlas history and where we're going, I'm going to jump more into some of the nuts and bolts of how to set up an account and how to figure out where these different survey areas, the Sentinel sites are, and then how to select a site within those larger polygons where you actually will go out and conduct a survey. So there are 2 separate websites that if you've been part of the Atlas in the past, you've probably already spent quite a bit of time on. The first one is Bumblebee Watch, and this is where you'll do all things data. So you'll enter your data here. You can see past records that you've entered, and then you can also go back and look at your own data and data that other people have entered. So if you want to explore bumblebee sightings across the state, you can do that on the maps here. The other website is the bumblebeeatlas.org website that Rich mentioned. And this is kind of a brand new reincarnation of that website that's really great, has a bunch of resources, and is easy to navigate. So I'm going to go back and we're going to jump over to a live. We're going to actually go to this website so we can look through it together. Rich, just to confirm, are you still seeing, you're seeing the Bumblebee Watch website?
Yes, it looks good. We're seeing the website. Although if you want to like Control+ to make it bigger, your screen's so wide that it it's kind of yeah.
Is that nicer?
That looks better. Yeah, super. Thanks.
Okay, perfect. So this is the Bumblebee Watch website. You can create an account by going over here to the top right to create an account or to log into one if you already have one. So if you go here, you can sign in. Given this website was recently updated, if you haven't been on the new website, but you do have a login, you'll have to reset your password using this link here. And then the other option, you can register right here for a new account. And this is where you'll be entering your Bumblebee data. So this is kind of the first step of adopting a cell and being part of this project, is to set up your account on Bumblebee Watch. We'll then hop over to the Bumblebee Atlas website. I'm not sure, there we go. So this is the homepage of the new Bumblebee Atlas website. There's a ton of different resources that you can explore. It looks like this is still showing up a little small. Hopefully that's a little better for everyone. There are a bunch of resources that you can explore from this menu tab. I'm going to show you two of the main pages that you'll be looking at. So if you go to the Get Involved page, you can click Find Your Atlas. And you'll then go to Pacific Northwest. And from here, you can find a whole variety of information about the Pacific Northwest project, steps of how to contribute. There are a variety of resources and things. But what we're going to go to first is this grid adoption page. So that's where you'll check out all of our new Sentinel sites across the 3-state area and think about which one you might want to adopt, and then start planning your survey from there. So you can get there either by clicking this grid adoption page link here, or if you go to Get Involved, adopt a grid cell up here. And it may take just a minute to load. So in these before you get started links, you already went ahead and set up your Bumblebee Watch account. So you have that all set. Make sure you have appropriate permits. That's something we can come back to once you've looked at the map and thought through where you want to do your surveys, but we do want to stress that you need to have permission to survey in different places. So thinking through where there's public land within those larger Sentinel sites. So we're going to go down here to our grid cell map. This is just a little overview of how this map works. So there's a legend and layers. I'll just show you that. So we'll say proceed for now. So this is the map for all of the different Bumblebee Atlas projects. For Pacific Northwest, you'll just click here. And that will zoom us in, so Oregon, Washington, Idaho. You can then go up here on the top left. This is giving us the legend, so just showing us Pacific Northwest is in green. It's a little hard to see at this zoom level, but if we zoom in, you'll see so look under Pacific Northwest, you can ignore the California, Midwest, these other areas. But in purple, you're seeing the Sentinel site areas. Other things you can do on this map, briefly, I'm sure you've all used maps like this, but you can change the base map, which is kind of helpful if you want to see the topographic map. If you want the USGS map, you can choose what you want to see there. This button just gets us back to selecting states. And then in this area, this bottom layers tab, we can turn on and off layers. And you can ignore the southeast for now. That's for a different region of the country. But one layer that you might be interested in looking at when you're planning your surveys is the USA Protected Areas public access layer. So if we turn that on, you can now see different land ownership. So this is really helpful for deciding where you do have permission or could access. I
Well,
believe
you just show people if they actually click on that red polygon, it'll actually pull up sort of the land ownership, and they can learn about it. And if you scroll down, there's more.
And I think some of them have links, right?
Yeah, some of them do have links, but that's just, it looks red, so it may be like, oh, you can't go there. But I would just encourage folks to be sure to poke around and actually click on them, and then you can learn about who manages the land and what the access is like.
Yeah, so this is a great resource for learning more about where you can actually survey. I'm going to go back and just turn that off for now. All right, so let's say you're sitting around at home wondering where you are going to do a survey. So I'm based in Portland and I really like Mount Hood. So I'm zooming in on this survey site over here, and you'll notice that when you're zoomed out, you just see the polygons. Oh, if you're zoomed out far enough. And then if you zoom in, they populate with the number. So this is the code associated with each of those polygons. So the Mountain Hood area is OR6. So if we zoom in, you can get a better sense of the area that covers. I'm just going to switch to the imagery layer so you can see. So this is about a 7.5 kilometer diameter polygon made up of hexagons within it. So it covers, you can see it covers a really pretty large area. And within this site, you'll be able to pick the location where you'd like to do a survey. And we'll go over a little more of that later of how you would actually pick out a site that would be a decent place in there. But one thing you can do on this map at this point is if you keep zooming in, you'll see that these yellow dots appear and the purple goes away and you just see the purple around the hexagons. So if you click on one of these yellow dots, you'll see information about surveys that were conducted there in the past. And this pops up, this box. And since there are multiple layers overlaid on each other, this first one you're seeing highlighted that whole hexagon. So to see the actual survey, you'll go over here and click this little arrow, and then the box will come up and give you information about the survey conducted there in the past. So you can see date, bee richness, so that'd be the number of species. So this can give you a good sense of where within this larger polygon other people have already conducted surveys. So that's kind of a tip off that those could be good spots to go, both because of potential public access, although some people have conducted surveys on areas they got permission to go on, but may not actually be public. So that's still something to look into. But you do know that bumblebees have been found in these spots before. There are floral resources, at least at the date that they listed. So we'll say we're adopting this Oregon 6. So our next step is to go down to our grid adoption form. So to fill this out, you'll enter your name, first name, last name, your zip code, your Bumblebee Watch username. So if you haven't done that already at this stage, you'll have to go create a login. You'll have email address. And then once you go down here, you'll select what state you're adopting the grid in. So I did Oregon. And when you populate that, you'll get these other categories opening up. So one is the grid cell. So you'll find the grid cell you wanted. So let's see, Oregon 6 was the one we decided on. And then like Rich said, we're asking that people ideally visit the site 4 times throughout the season, separated by approximately 3 weeks. So this now shows you those different time periods. So this June 1st to 23rd, 24th to July 14th, July 15th to August 4th, and then this last one in August. So this part of the form is asking which of those dates you can commit to surveying. And ideally all 4, but we know people have other things going on, may not be able to make it. So even if you can only survey once, that's still really great and a good contribution. So don't be scared off if you can't survey all 4 times or even 2 times. So you'll go here and fill out, we'll say, yes, we can do these dates. And then if you fill out no for one, you'll get this new box that asks if we can share your contact information with other people. And that's because we'd like these surveys to be at the exact same location as the ones. So if you've done the first 3, we'd like someone to be able to go do that 4th one in the same spot. So you can say no if you're not comfortable with us sharing the information, but if you say yes, then we'll be able to get in touch and connect you with the person who'd be completing that time period at the same site as you. You'll then just fill out a few more boxes certifying that you're 18 years old. If you're not, you can still do this. You'll just have someone over 18 fill this out and be involved in the survey with you. You'll fill out our liability, saying you've read our liability release form. And then this last section is the trespass policy. So confirming that you know that you can only survey on public land and that these polygons straddle a whole bunch of different land ownership. So you'll, in planning, need to think through where within those polygons you have permission to survey. then the last one is acknowledging that you are a volunteer and won't be paid. And then there's one other thing I want to highlight going back up, which is this link. So under, when you say Pacific Northwest long-term monitoring, there's this link that says visit this page to learn what other grid cells have already been adopted and what time periods. So if you click on that, it will take you to this dashboard. And this is where we've recorded who has signed up for which survey sites. So you can see the station ID over on the left and then the 4 time periods here. And then the number within each of those cells shows how many people have signed up for that area during that time. So we're really hoping to get as many of these areas adopted as possible in all the time periods. So if you're kind of waffling between 2 different sites, it'd be great if you come check this out and adopt one that hasn't already been adopted. Or if you're, for example, in this area where Idaho 4 is, and you could do it in this time period that's empty, you could sign up for that. There's not a problem with doubling up. The more data, the better. So if there's someone already signed up, but you really want to go to that site, feel free to still sign up for it. We also know that sometimes things come up, so you might sign up for a site in a time period and then you're not able to actually go do the survey there. So we just ask that if that happens, you reach out to either me or Rich, just shoot us an email and let us know, and then we can see if we can get someone else to cover that site. And then, so we're back to the main page. Once you submit this form adopting a site, you will then get a confirmation email letting you know which sites you adopted. And submitting this once only lets you adopt a single site, but if you'd like to adopt multiple separate areas, you'll just come back here and submit the same form again. Same information up here, and then just entering the different grid cell or the different long-term monitoring ID in this area. If it pops up. I think if I delete this, you will, yeah, you would get your full list again. All right, I'm going to hop back now to our presentation quickly. Are you seeing that again, full screen, Rich?
Yep, looks great.
All right, so we just went over this, the page on the Bumblebee Atlas website for adopting your survey station. So the next thing you'll need to do once you've adopted it, you've decided on a site, and then you've officially adopted it on the website, is thinking through a little more of the fine details of how to plan your survey. So a key step is deciding then within that polygon where you have permission to survey. So some ideas of the best land ownership to look for that we do have permission for is U.S. Forest Service and BLM. And then some other options are state parks, state wildlife areas, local parks, natural areas, or roadsides. And we don't guarantee that these do have public access, so make sure that you contact local authorities or people in charge of those areas to confirm that you can access them. And within those, we also really want to be focusing on natural areas, like natural habitat. So if your polygon covers a giant parking lot, we probably prefer that you go to somewhere with a little more natural habitat than a parking lot. So while you're thinking that through, it's good to consider where you're going to see habitat that might support some of these more rare species. So as Rich said, we're looking to gain more information about specific bumblebee species over time at a location, and that's a change from previous years. So to fully understand what species we're seeing at a site, we want people to survey across the whole flight season of bumblebees. And that's why we've laid out these 4 different windows that we'd like people to survey. So on these calendars, you can see what those are. They're June 1st to June 23rd, June 24th to July 14th, July 15th to August 4th, and then August 5th to 25th. And our main goal is to have those surveys about 3 weeks spaced. So we hope that people will keep a few weeks in between each survey and not, for example, survey on June 23rd and then again on June 24th and count those as 2 separate time periods. And like we said, if you can commit to all 4 time periods, great. If you can't, that's also great. We're really excited about people's involvement, and even doing a single survey is really helpful. Surveying conditions, when to survey. Basically, if it's a day that you want to be out having a nice time outside, then it's probably a day that the bumblebees are going to be out too. So the weather to shoot for is temperatures generally between 60 and 90 degrees. You want pretty calm winds. Bumblebees will fly in some wind, but generally below 15 miles an hour is a good thing to shoot for. And then no precipitation is ideal. There can be some cloud cover. Bumblebees are pretty hardy flyers, so if there's a little cloud cover and a little wind, it's not that big a deal, but you do want it to be warm and not howling wind. So once you've selected a survey station and you've just kind of narrowed it in on an area and you're going there, there are a few things to think about for a specific 1 hectare area where you're going to be conducting the survey. And the goal in this is to select high quality habitat where there'll be bumblebees throughout the summer, and ideally the target species. So here are a few ideas of what to look for and how to decide on areas to focus. So you're going to be looking for a site that has a variety of different habitat types within it that would support different species and provide resources across the entire summer. So something with a whole variety of plants would be great. If you have an area that's riparian or wet in one part and dry in others, things like that are really helpful to look for. Try to pick a site that has some flowers, but also looks like it will be flowering throughout the summer. So you don't want a place where everything's in bloom right when you go, but in a month or 2, it will likely all be dried out. So again, looking for a variety of species and at different phases of flowering. And then here's a list of questions that you can ask yourself when you first get to a site to see if you think it's going to be a good site to survey. So do you see plants in bud that have not yet flowered? That's a good sign because you'll be having flowers blooming for the next at least couple weeks, hopefully a month or more. Is your site currently wet but likely to dry out? You don't really want a site that the whole thing's a bog to start out with, but having some wet areas can be good for making sure that you have water to have plants blooming later in the season. Is there a permanent water source? And that could be from melting snow if you're at fairly high elevations. That's another good thing to support these plants blooming throughout the summer. Are there flowers already going to seed? This could be a bad sign if all of the flowers are going to seed, but if it's just some of them and there are other species blooming, then that may be okay. Just pay attention to these first 3 bullet points if you are seeing flowers going to seed. Are you still seeing some queen bumblebees? That's a good sign. That means colonies are just getting initiated and you'll likely have bumblebees for quite a while. And then are you seeing any males? That's probably a bad sign since males are produced near the end of the colony life cycle. So that means it may be winding down and you'd see fewer bumblebees. We kind of covered this, but this is just reminding you of the resource online, looking at that map to select a good spot to survey. So again, just letting you know that you can go to that map, zoom in on one of these larger survey areas, and then continue zooming in onto one of these survey sites and checking out using this little box what those survey sites are. So if you're looking from a ways away and thinking about where should I even go look to begin with, this is a great place to start. These overall areas were selected because they had places that we'd surveyed in the past, and we think it's likely that we'll find some of these focal species since we have found them in the past. So this is a really good area to start, is thinking about where surveys have happened and maybe checking those out as your site. When you've narrowed it down, you're ready to go out in the field. This is just a list of materials that it will be helpful to bring with you. So data sheets and pencils, we have data sheets on the website that you can print out at home and take with you. And insect net and vials. And in just a minute, Rich is going to go into how you're going to use these all. So don't worry too much about that, but this is kind of the list, which is also available on our website to look back at, but of what you'll need to bring. You'll need a cooler with some ice in it, a camera or some way to take pretty high quality pictures. So most smartphones have a good enough camera for this. A GPS unit, or a phone with mapping apps or paper maps, just some way to navigate to your site. A timer or a smartphone with a timer so that you can keep track of how long you've been surveying. Some way to know weather, to have a sense of what the weather is going to be like. It can also be helpful to measure temperature and wind speed at the site. And then if you have any field guides, you'll be asked to be identifying plants if you know what they are, and it can be helpful for identifying bumblebees as well. So if you have field guides for either plants or bees, those are great to bring. We touched on paper maps, and then bringing friends or other people is a great way to have a fun day and to make your surveys a little more manageable, since we're doing surveys for a certain number of minutes. So that's people minutes. So if there are 2 people, you're surveying for half the overall time as 1 person. So it's a great way to bring other people out. And I think now I'm going to hand it over to you, Rich, to get started on how to actually conduct the survey once you've selected your spot and have all of your materials.
Okay, great. What we're going to do right now is actually, a number of years ago, we created a video that I've recorded outside. This was done during COVID when we couldn't have in-person events, and we were recognized that folks really needed to get sort of hands-on experience to learn how to do these surveys. And so we tried to adapt. We started doing these virtual trainings and recorded this video. It is about a half hour long, so we're just gonna play it and let you watch it during the webinar here. And I will also just let you know that this coming Saturday, Molly and I are going to be doing an in-person workshop. It's in the Portland area. If you are near the Portland area and want to, get a refresher or see the new protocols in person, or you just want to meet Molly and I and say hi, we welcome you to join us. I believe it's 10 a.m. at the Tualatin National Wildlife Refuge. There is a registration link. It's on the website. If you go to the events page there, you can find a registration link. We can probably drop a link to it here in the chat as well. But I would encourage you to come out. If you have any trepidation, I would encourage you to come out and try to join us. I realize that it's a long way to travel for many of you, and Portland is a very small area in the Pacific Northwest, and we wish we could sort of go on tour and do a bunch of these, but that's just not in the cards for this coming spring. We may be doing an additional workshop somewhere in July, but just stay tuned for that. And yeah, if you have the opportunity to join us in the field, we definitely welcome you. All right, I'm gonna see if I can share my screen back over you here. And
great, and I just dropped a link to that field day.
Great.
Into the chat.
Thank you. And Molly, can you see my screen?
Yeah, looks good.
Okay. Hopefully this will play. Hi, my name is Rich Hatfield. I'm a senior conservation biologist for the Xerces Society, and I'm here to teach you how to do a survey for the Pacific Northwest Bumblebee Atlas or the Nebraska Bumblebee Atlas. They both have pretty much the same survey protocols. So I'm here today. I'm in a beautiful natural area. It's a nice, warm, sunny day.
I've got my net and I have some vials in my pocket, so I'm pretty much ready to do a survey. The first thing you want to do is make sure that the site is big enough. It needs to be about a hectare. And a hectare is about the size of a football field, lengthwise, square. So sort of a football long and the same distance wide. It's 100 square meters, so a football field, including the end zones. Another way to think about it is the infield of a track. If you ever ran track, the entire infield on the inside of a running track is about a hectare, or a soccer field is also about a hectare. So you want to make sure it's big enough. And then usually what I do once I decide that it's a big enough site is I walk around and I make sure that there's enough flowering resources. You want to make sure that there is something that bees could be visiting. Otherwise, it's probably not worth really conducting a survey, or maybe come back at a later date when there are flowers. One of the things you want to do when you're out pre-walking your site is make sure you're keeping an eye out for inconspicuous flowers. Sometimes flowers will grow down amongst the grass, but they can be important for bumblebees and often are. This plant is called self-heal and often grows underneath the understory or overstory of grass and is a great bumblebee plant. Another important thing when we're out looking for bumblebees is we want to make sure we're looking at all blooming plants. If I was standing here looking in this patch, I might be drawn to the violets, to the dandelions, or maybe to the bluebells over here. But in reality, one of the most attractive plants in this patch has green, inconspicuous flowers, but bumblebees love it. This is a plant called waterleaf, and the flowers aren't quite open yet because it's still very early in the season here. But these little green guys here are going to bloom into little, beautiful little flowers that hang down sort of under the understory, and they often can't be seen. But bumblebees like Bombus flavifrons and other long-tongued species really love these flowers and will visit them. So while you may not be drawn to them with your own eyes, it's really important to pay attention to what's blooming and making sure that we're spending time looking at all the flowering resources on the landscape, looking for different species of bumblebees. Other things you may want to look for as you're doing a pre-walk around your site are things that may be a potential hazard. You may want to avoid plants like poison oak or blackberry, although it is notable that both of these plants do flower and can be attractive to bumblebees. You'll also want to look out for holes or other things to avoid, like electric lines, fences, barbed wire. Just make sure you're safe while you're doing your survey. You also want to keep an eye out for property lines to make sure you're not crossing from public land onto private land. And also keep an eye out for areas that you may want to spend a couple more minutes, areas with high quality forage that you know are especially attractive to bumblebees. Or keep an eye out for blooming trees, meaning don't forget to look up. There are sometimes flowers that are above your head that can also be attractive to bumblebees. Once you've finished your pre-walk, you're ready to start your survey. Materials that you want to make sure that you have with you before you begin your survey are an insect net, jars to place your bumblebees, a cooler with crushed ice. Make sure you have a timer. And then for processing your bees, you'll need your data sheets as well as a camera. We recommend having a dedicated camera, ideally with a macro function, but in a pinch, a smartphone can work as long as you learn how to control your white balance and take high quality photos with it. I typically set up a station in the middle of my survey area, which has the cooler and the area where I'll process the bees later. And then I grab a handful of vials and head out to go conduct my survey. And when you're conducting your survey, you want to make sure it's 45 minutes long, and that's 45 person minutes. So if you're doing it with a friend, you would do it for 22 and a half minutes. And if you did it with 2 other friends, it would be for 15 minutes. You just want to have 45 total minutes of search time. The important thing also is that when you're doing a survey, you want to pause the timer while you catch a bee. So it's 45 minutes of searching, not 45 total minutes. So you want to pause the timer when you catch a bee, and while you're putting that bee into a vial, and while you're putting that vial into the cooler. That doesn't count for your search time. The other thing that's important while you're doing your survey is you want to make sure that you're associating every bee observation that you have with a flower. So one of the important things about the Atlas project is we want to learn what resources that bees are using. So while you're out collecting bees off of flowers, you want to make sure to record that information so that we can gather it later and use that information when we analyze the data from the project. So with each bee that you capture, you want to also record the flower on which you captured it. Catching a bumblebee in a net is not an easy task. It takes a lot of practice. So one of the things that we recommend is that you spend some time practicing in your backyard before you go out and actually conduct your survey. That way you'll get some competency and some confidence so that when you see a bumblebee during your survey, you'll actually be able to net it. There are really two different ways to go after a bumblebee on a flower. One way is to just swing your net and try to catch it in there like this. The important thing is after you've swung, you want to make sure to turn your net over to close the bottom of it so that the bee doesn't come flying right out into your face. There's a number of ways to do that. I like to do a little flip, but the easiest way to do it is to swing and then just turn the net over 1-2 times. And then you'll have the bee in this section of the net that's laying over the top. The other way to go after a bee instead of sweeping is to just go over the top and drop the net on the bee like that. That way the bee will be trapped in the net. The next step then would be to grab the bottom of the net and pull it up to the top, and the bee will fly right to the top of the net because it's attracted to the sun. Let's see if we can find some bees to practice on. Sometimes you just have to go and follow for her a bit. She looks like she's looking for a nest, so they can be really hard to track. But we can just throw the net over the top of them like that sometimes while they're flying. And I got her. I can see her moving in the net there. So what I do is I pull the net then up to the top and make sure she's under there. I see her right in there, and then I can put her in the vial. Okay, so now that I have a bee in the net, what I need to do is get the bee from a net into the vial. And the way I'm going to do that, she's somewhere in this folded over area. I can see her crawling around right here. And so what I want to do is make sure she gets all the way down to the bottom of the net. So what I can do is I can just take the net and just go back and forth a couple of times and then fold it over, and then I like to grab it when I know she's above my hand here. So I know that she's between my hand and the bottom of the net. And then I can take my jar, take the top off, but I want to keep that cap close by. And so then what I do is I stick the jar into the net, and it goes through my hand here, okay, so that there's no way for the bee to get out. And then what I can do is just sort of move the net around until I'm sure that I have the bee in the net. And unfortunately, my net is very dirty, so it's hard for me to see. But I can hear her. I know she's in there. And usually you'll hear that sound when you know she's in your jar. I know that she's in the jar. And now I can just use the net basically as the top of my jar. So I'm just going to reach my hand in, and again, I'm going to grab that plastic vial, and I'm just pulling the top of the net over the top of the jar so the net is still serving as the top until I get to the edge, and then I'm just going to slide it directly into and put the cap directly on, and then I can close the top and I can see the bee there in the jar. And that is how you transfer, or at least one method for transferring a bee from a net into a jar. Okay, so now I want to show you another method for getting a bee from a net into a vial. You can see the bees crawling around in the vial right here. And I'm sorry, in the net right there. And I want to get her into the vial. So one thing is a good thing to do. Some people just aren't comfortable holding the net while they do this, so they like to put it on the ground. But you can see if I hold the net straight up, the bee will fly all the way to the top of the net, okay? And then I can move forward. So some people just like to set it on the ground and then use the ground sort of as the cap, and then stick the vial into the net. And then again, same principle, you can pin the bee against the net as the top, and then you can stick your other hand in there and just put the cap on, and then you'll have the bee in the jar. Simple as that. And then what we want to do is we want to take the bee once it's in a vial, and we're going to put it into the ice pack. And it's ideal to stick it as far into the ice as we can so that the ice is completely surrounding the bee, and then it'll chill very quickly so that we can take the photographs that we need to take.
I'm just gonna pause this for a second just to talk about safety here. We've learned over the years since we made this video that sometimes those blue ice things that you see in here, not the ones that I'm using here, but a lot of the newer ones that are made by some of the more expensive cooler companies, when you chill them in coolers or in a freezer, in a cold freezer especially, they can get very, very cold, like well below 0 degrees Fahrenheit. Those temperatures can actually be dangerous for bumblebees, and we would ask that you try to avoid using them, or if you use them, to use them at the very bottom of your cooler and not as the primary source, or not put the vial actually in contact with them. So use them to help keep your ice cold, but don't actually use them to chill your bees. Likewise, if you have like a chest freezer that gets down to negative, 30 or whatever, try not to use ice from those coolers. The temperature actually just gets too low for bees. So, if you get ice from a gas station or from a regular freezer, those are fine. Those cubes are fine. But we have had, and to be honest with you, the mortality hasn't been so much in the Pacific Northwest, but in California, I think where temperatures are a bit warmer, the cold from the warm has been a shock to some bees, and we've had a handful of mortality, like 4 or 5 bees, maybe a few more than that, but not gross numbers. But just thought I would share this little bit of information about safety that we've learned since I made this video. So use crushed ice primarily, and not from a cooler that's too cold. I'm going to continue the video now.
Normally, one of the things that I would do in this situation is I would take the flower that I captured this bee on, and I would put it into the jar so that later when I was processing my bees, I could know which flower that I caught this bee on. And keeping track of that is very important for the data we're collecting for the Atlas project. Or in this case, because this bee wasn't visiting a flower, it was just flying around, it's not going to have any floral association. So if I don't put a flower in there, then I'll know that when I process my data later. But if I'd captured this bee on, say, a dandelion, I could come over to a dandelion flower and just grab the flower off and pretty easily just tuck the flower into the jar with the bee. And then later when I process this bee, I'd have the flower and the bee in the same jar so that I can keep track of it. Other people like to take notes and have a notebook, and they will number their vial and say this would be bee number 1. They could go bee number 1 and they would write down the flower, or take a picture of the bee next to the flower. There's lots of different ways to keep track of it. We don't care which way you do it. We just care that you do it. So when I finish my survey, I like to get a station set up to take my photographs. I want to make sure that I have everything I have and everything I need before I get started, because when I take the bees out of the cooler, they're going to start warming up, and I want to make sure that I have everything. So I want to make sure that I have my data sheet and a pencil that has lead in it. So I want to make sure I can take my notes. And I want to make sure that I have my camera. The other thing I like to do is make sure that my camera is set so the pictures I'm going to take are appropriate for the lighting conditions. So I usually will take a cap of a vial or something like that and make sure that I have my camera in the right settings before I get started, so I don't have to wait while the bee is warming up. So I get it in my macro setting and make sure that the pictures that I take look good. So here I am, I'm going to take a picture, and that looks pretty good. You can see here it looks pretty good. I could probably add a little bit of light. It's a little bit dark, so I may change the white balance a little bit, but for the most part, it looks pretty good. So once I'm ready and I've got the information that I need, I can start processing my bees. But first I want to make sure I've filled out the top of my data sheet. So I have my site name and my grid cell ID in here, along with the date and the survey method, my weather information, and the surveyors and the survey time. All of that's entered on the data sheet, and I can get ready to enter the bees. Now each line on your data sheet here is going to represent a vial. So each bee that you captured gets its own line. So you're going to enter what bumblebee species that you think it is, its host plant, and then the photo numbers that you need, that you took of that bee. I'm going to move and process my bees in the direct sunlight here because it often will take a different picture, or a better picture, if you have direct sunlight. Sometimes it's too bright. You just have to take a look at the pictures that come out of your camera and adjust your settings and your location appropriately. And I can't stress this enough. I get way too many photos that people took that are out of focus, or are too bright, or are too dark. And if someone goes through all of this trouble of collecting bees, puts bees in jars and bees in the jars in a cooler, and then takes pictures that doesn't allow me to identify them, a lot of that effort has been wasted. So taking pictures is really the most important thing that you can do. And I want you to make sure to get your settings right and take the time you need to get the best pictures that you can. So I'm ready, my station's all set up, I have everything I need. I can grab my first bee out of the cooler. And once you take a bee out of the cooler, you'll know she's ready to be processed if you can kind of shake the jar back and forth, and she moves pretty freely in there. That's when you know she's ready to be processed. And so usually what I do is I'll just dump her out onto some surface, and if you have white, that's great, it works really well. Other colors can work too. Darker colors, just something that's flat and neutral usually helps pretty well. So one of the things that we can do when we look at our bee, remember there's a lot of different photos that I need. I need to be able to see the picture of the face. I need to be able to see a picture of the thorax. And I need to be able to see a picture of the abdomen, including all of the segments back there. And for yellow-faced, yellow-striped bees, I need to see a picture of the underside of the abdomen. That allows me to tell the differences between some very close look-alike species. So when they're completely out is a great time to take a picture of that underside of the abdomen if you have that species. And in this case, I do. So I want to get really close if you can, and this is why a camera with a macro setting is really the best option. You want to get as close as you can, and you want to take a nice picture of the underside of the abdomen. Okay, so now I can see the color of those hairs on the abdomen, which is exactly what I need to see. Also, when she's in this position, I can often take a very good picture of the face. And it's nice to take a nice close-up like this, because then I can see the cheek length, as well as the color of the hairs on her face, and the colors of the hair on the top of her head. I also like to include a profile shot like this. Okay, And one of the nice things about a shot like this is I can see the hairs on the face, on the thorax, and on the abdomen. And now I want to be able to make sure that I can see every single segment on that abdomen and then the color patterns there. So you want to sometimes peel those wings apart and make sure you get a nice clear shot. It's okay if your hand is in there, but we really just want to be able to see all of the segments back there on the abdomen. Perfect. And then she's ready. I've taken all of the photos that I need for her. You can see there that her foot is starting to twitch a little bit and she's starting to wake up. But I've taken all the pictures that I can of her. And so what I can do is I can actually just set her in the sun and let her warm up, and she'll slowly warm up and groom herself and fly away. And then I can go on and process the next bee. But before I do that, I want to make sure I enter this one on my data sheet. So because this has a yellow face and a yellow stripe, and you notice that all the hairs on the underside there were black, I know that this is Bombus vosnesenskii. And if you don't know the species of bee that it is, it's totally fine. You can just write Bombus here. And I'm going to write the host plant. In this case, there is no host plant. I found this bee just flying around, probably looking for a nest site. So I'm just going to write nest searching. And then I want to write the photo numbers. So in my camera here, I'm going to open it up, and I'm going to open up, and I'm going to take a look at the photos that I took. And down here, it's going to tell me the number of the photo that I took. And I want to record that information on the data sheet. So these photos started at 2319, and I'm going to write that on here, and that goes through 2324. Okay, and now that's all the photos that I've taken of that bee, and I've entered all the information that I need to from that particular bee on the data sheet. Now I can process my next bee. Again, I can shake the bee in the jar, and if she moves around freely, I know that she's ready to be processed. I can dump her out onto the table. And again, this is not a yellow-faced, yellow-striped bee, so I don't need a picture of the underside of the abdomen. I just need a picture that shows the face, head, thorax, and abdomen. And I can get a lot of that from a single shot with this bee, the way that she's set up. I can get a nice profile shot. And there I can see pretty much everything I need to from a single photo. I'm gonna roll her over on her back to make sure that I get a photo of the cheek length and of the face. Sometimes the antennae get in the way there, so you wanna make sure, if you can, to get the photo that you need. Okay? So there, really, with just two pictures, I can get everything that I need. But there, in this case, I can also get a nice shot there that'll show me every segment of the abdomen. And this species here is Bombus mixtus. It's got brown hairs at the very back of its abdomen, that little crescent on T2 there. And you can see it's got cloudy hairs on its thorax. So a mix of yellow and black hairs and a nice short cheek. So I'm done processing that bee, and I can put her in the sun as well. So now I can write this one as Bombus mixtus. She was also nest searching. And the photo numbers, again, I go into my camera, I press preview, and this goes through 2327. So this is going to be 2325 to 2327. One of the things you can do when you're done processing your bees is you can actually hold them in your hands. It's a really neat opportunity to get up close and personal with a bee and see how they move their wings and groom themselves. It's a really, really neat opportunity. They do warm up pretty quickly, and it's pretty neat to watch them fly away and notice that even after being in a cooler for a long time, they can go out about their business and just fly away. Oh, there she goes. When you're done with your bumblebee survey, we're going to ask that you also do a brief habitat survey. And there's just a series of questions that you have to answer as you walk around the site. There's some just basic site information at the top of the data sheet, and then we get into the habitat area. The first thing you want to do is decide what is the most appropriate habitat type for the place where you're doing your survey. And you're just going to circle one of those options. So in this case, I'm in a natural area, which is a park setting. So I would circle that. And then you would want to add in.
I think I am going to pause the video here because enough of this has changed that I think I'll probably just walk you through the data sheet, the new data sheet that we are going to be using instead of making you watch the
habitat area of the surround.
And then also walking you through that as well. So Molly, I've switched to a different screen. Can you just let me know if you can see the data sheet here?
Yeah, I can see the data sheet and like you can see the tabs at the top of the screen too, but you can still see the data sheet well.
Okay. See if I can get to full screen here. There we go. Is that better?
Yep, that looks great.
Okay, great. Yeah, we're going to have you do a brief habitat survey, and I'm going to walk you through the different parts of the data sheet now and the kind of information that we are looking for. Most of this is going to be identical if you've participated in the past and done surveys. There are a few places where that is different, and I will try to highlight those differences as I go through the data sheet for those of you that are returning. So in the top, you're going to fill out the same information, the site name, the site ID number, so the grid cell that you've adopted or the Sentinel site that you've adopted, the date, the latitude and longitude, and you can find this using your smartphone if you open up Google Maps or Apple Maps and do sort of a long press where you are, it'll pull up the latitude and longitude. Even if you don't have self-service, your phone will still have a GPS chip and it should be able to do it no matter where you are, even if you don't see a map underneath your location. You'll fill out the number of observers that you that are in your survey. You can use as many as you want really, and then put the observer names or initials here. And then generally speaking, for this new protocol, we're going to ask that everybody do point surveys. So you generally won't be circling roadside here, but if you are, while the priority for us this year is to do these surveys in Sentinel sites, we do want folks to feel comfortable, like if you can't get to a Sentinel site or you want to keep surveying a place that you have in the past, we also encourage you to do that. Like surveys anywhere are welcome. It's just that our priority this year is to do these Sentinel sites. So if you want to know how to help best, it's to do one of these Sentinel sites. If that's not possible for you, you can keep doing surveys as you have in the past. So you'll enter the survey type, and then you'll enter the survey area. All of these should be 1 hectare. The Bumblebee Atlas surveys should be a single hectare. And then there's this somewhat new question here that says, I captured all bees, different bees, or used a combination of methods. In the past, we've really encouraged folks to capture all the bees that they found and to use that method. Now, what we're really asking folks to do is to really catch for diversity. We want to try to get as many different species as we can. If you're new to bumblebee surveys or you still don't feel confident at all in your bumblebee identification, or you have a hard time telling species apart in the field, you can continue to capture all bumblebees. That's totally fine. But if you feel like you're fairly confident in your bumblebee ID, instead of catching 45 Bombus bifarius in the mountains of Idaho, we would like you to sort of look for diversity and try to catch more different species. We still want Bombus bifarius represented in the survey, but we don't need 30 of them. But if you don't feel confident in that, err on the side of including too many bumblebees. That's just totally fine. But here you will note which method that you used, whether you might start catching all of them and then realize you're getting a lot of the same, and then switch to collecting ones that are different, and then you might select combination here. You're going to note the start time and the end time of your survey. Note that this is an hour here, right? But my survey minutes is actually 45. So all surveys should be 45 minutes. This took longer because while I was, capturing my bees or putting them in jars or in the cooler, I paused my timer. So that's why the survey start time and end time is different than the survey minutes. But the survey minutes should really always be 45 here. Then you're going to get general weather information about the temperature and the cloud cover and the wind speed. You can get temperature and wind speed later. Like when you get home, you could use a weather app at home to get, the weather conditions near where you were surveying if you don't have that information and can't get it directly while you're in the field. And then you're going to look at the habitat information for the survey. A lot of this is the same. So at the top here, you're going to circle, what habitat type that you're conducting a survey in. And then over here on the right, you're going to note up to 3 habitats that are surrounding your 1 hectare. So the habitat type here is the hectare in which you're conducting the survey, and it would be the dominant type. So you might be conducting a survey in a hectare that has some woodlands and some shrub and some grasslands, but you're going to choose the most dominant habitat type here. Are you in a grassland? Are you in a riparian area? Or are you in a forest? And then over here on the right, you're going to choose up to the 3 most dominant surrounding habitat types. It's fine if there's just one. You might be surveying in a grassland hectare that's surrounded by grassland. And you might just answer grassland here. That would be totally fine. Up to 3. And then you're going to choose here, you're going to, just like in years past, you're going to choose how much of the survey area had flowering resources available. It's important to realize that this is not % cover. So we're not asking you to estimate how much of the hectare had flowering resources, like looking from above. It's more in how much of the plot could you search for bumblebees foraging on blooming plants. So even if the plants are separated by a couple feet, like you're still looking at those flowers, that would be included in that estimate. So it's really only areas that are sort of bare and have nothing in bloom that would be excluded from this question. And then you're going to choose the following features that you see in your survey area, whether you saw bunch grasses or rock piles. These tell us about bumblebee nesting. And then you're going to answer some management questions. Do you see mowing in the area? The livestock grazing question is new. So in years past, it's just been yes, no, or suspect. We're going to try to get more about the degree of grazing at sites. So we're going to ask you to circle whether you see any of these 4 features of grazing in your habitat plot or in the survey plot. Do you see stubble, areas where cows or other livestock, and we're not talking about native grazing here, it's just livestock grazing. Do you see stubble? Do you see hoof prints? Do you see cow pies? And do you see animals present? And you're going to circle every one of those that you saw while you were doing your survey. Insecticide, herbicide, fire, yes, no, or suspect. And then this is going to be another new part for the long-term monitoring protocol. Instead of just noting whether you saw honeybees, we're going to actually have you walk around for 10 minutes after you have completed your survey and count the number of honeybees that you see. So you're just going to set a timer for 10 minutes, amble around, walk around your plot for 10 minutes and count the number of honeybees that you see. And you're going to note that on the data sheet here. In this box down here, you can write any notes that you had about management or about the site, and you can include those when you submit your data as well. Then you're going to look at how many different species of flower were in bloom in your site, and you're going to note that here. Then on the back, you're going to list those plants. So you can see that here I listed 3 plants were in bloom. And then on the back, what I'm going to do is I'm going to list those plants to the best of my ability. Ideally, you're going to get to species here, but you may only be able to get to genus. There are a lot of resources that can help you with plant ID. Taking photos is a good one. Seek from iNaturalist is a great resource for identifying plants or at least getting to genus. And you can also use the Facebook group and other resources to try to have help identifying some of those plants. And then in addition to just naming those plants, we're going to have you try to estimate % cover this year. So in this plot, I said that there was 60% of the area was flowering, but now I'm asking you how much of that 60% was made up of these three different plant species. So I said that 60% was Potentilla, 25% was Lupinus, and 15% was Pedicularis. I made this up just as an example, but we're going to ask you to do this for the top 10 blooming plants in your plot, plus any plants that maybe weren't in the top 10, but were visited by bumblebees. So if you had I don't know, maybe there was one Phacelia blooming in one corner of your plant that doesn't make the top 10 list, but it was visited by bumblebees, we'd like you to list that here as number 11 and include it like as 1%, as a minority of the plot. And then you're also going to enter the bumblebee information on the back of the data sheet, just like we showed you in that video. The one thing that's going to change this year, and again, this is ideal. It's okay if you can't do this, but one of the things that we're trying to learn is how long does it take during your survey for you to find one of our target species? So you'll see here in my survey that I found Bombus occidentalis, and I noted that at 111 was my time of first detection. So when I captured this bee at 111 and put it in my jar, I looked at it in the jar and I said, oh gosh, that is Bombus occidentalis. And so what I did is I put a little note, or however you want to do it at this point, but you could write, on the jar or put a note in a notebook that said, B14 or whatever number you have on your jar there, the first detection for this was this. And then when you enter this in your data, when you're processing the bee, you're going to enter this over here on your data sheet, and then we're going to include that information when you submit the data. This will help us understand whether a 45-minute survey is too long or too short, and it will help us understand how much effort it takes in each survey to detect our target species. So you're going to do this for Bombus occidentalis, and then also for Bombus morrisoni. And again, if you're not confident with your bumblebee ID and you can't do this, that's fine. You don't need to be able to do this. This is just for those folks that have been doing this for a while, that know those two species, that could help us learn a little bit more about what it takes to find them at plots. Don't let this intimidate you. This is just something that helps us. And then at the bottom of that page, you'll just fill out the volunteer data as you have in the past. So that's the habitat survey. There isn't a whole lot that's different here between the old protocol and the new protocol. We're adding in the percentage of plants, the the livestock grazing, and the honeybees. Those are the three new pieces that you'll be doing in your new habitat survey within our long-term monitoring plots. So to fully hammer this home, the key differences from previous years, we're now focusing on looking for bumblebee diversity. So we're trying to find every species of bumblebee present at the site. If you have like Bombus vosnesenskii and you suspect that another lookalike like Bombus caliginosus might be there, then we ask you to capture 10 lookalikes, and then you can focus on finding bees that look different. We're noting the first detection of Western bumblebee and Morrison's bumblebee, as I showed you. And you're going to note that on your data sheet when you enter it. And then after your survey, you're going to count the number of honeybees that you find in a 10-minute walk through your plot. It's fine if you don't make it through the whole hectare. Just walk from flower to flower and see how many honeybees that you see. And then you're going to note that on your data sheet as well. And then finally, you're going to document the plant community. You're going to list the top 10 plants that make up the flowering plant community and estimate how abundant each of those are using a percentage. And those percentages should add up to 100. So how much of the top 10 plants does each of those make up? And just to give you some idea of what this would look like, if up here on the left, if this is what my plot looked like, I walked around, these were the different flowers that I saw. I had areas that were, occupied by flowers and areas that won't. So over here, I counted the number of blooming plants. There are 9 different species of blooming plants in my plot. And if I sort of in my brain, I try to move them around and figure out how much of the plot that they, take up, this seems to be around 50% of the plot was areas that I could look for bumblebees in, and 50% was just sort of empty space. So I would write that on my data sheet as 50%. And then again, in my brain, try to think about, how much did each of those individual plant species make up in terms of the total bloom? And here I estimated, that these were 5%. This was a little bit more than that at 8%. This was the most abundant at 40%. This was maybe half that at 20%, and then fill that out so that it adds up to 100%. Just do the best you can. This does not have to be perfect. It's not an exact science. We're just trying to get an idea of not just what plant species are blooming, but the quantity at which they are available. And then you'd note this as I showed here on your data sheet. So you'd note the plant species, and the percent of the bloom on the data sheet. For photography, I think I went through this pretty well in the video, but I just want to stress that taking pictures is the most important thing that you can do. Everything else is kind of superfluous. Like if you can focus on taking great photographs, that allows us to get the best identification that we possibly can. So if you're going to, travel X number of miles to a site and spend X number of hours, please try to get good photos so that, all of that great effort, not that it wasn't fun to just go and that's worth it too, but we'd also like to make sure we get, data from that visit. So take good pictures. We need to see the front of the face, the hairs there, the top of the head, the vertex, and then also the cheek length, the area that you can see in red here. Those are all important photos to take of the head. And then on the thorax here, we want to be able to see, what's behind the wings, what's between the wings, what's in front of the wings, as well as what's below the wings. The hair color in all those areas is important. And then we also want to be able to see the abdomen, and we need to be able to see really all of the different segments there, including the middle. So we want to make sure to peel those wings apart and get a photograph of underneath the wings there. And again, like we've stressed this number of times, but like make sure you're taking high quality photos. A good, clear question for yourself is if when you look at your photos, like on your camera or your phone, if you can see individual hairs on the bee, it's a good indication that you've got a pretty good photo. If you can't see or that's blurry, then try to retake those photos. And yeah, if you need to part the wings or get your fingers in there, no problem. They don't need to be National Geographic photos. They just need to let us see the characteristics of the bee. Okay, so now that being said, what I'm going to do now is just walk you through the new protocols for submitting data. And I'm going to just change the way that I'm sharing my screen here, because I'm going to, let's see. This is not doing what I want it to do. Sorry, bear with me here. I'm just going to stop sharing. There we go. Then I will set this up so that you can see it as I want you to see it. Okay. So I'm going to start sharing my screen again. I will share the whole screen too. You should now be able to see my whole screen. So when I get ready to submit my data, what I want is you open up Bumblebee Watch and make sure that you're signed in. And then you want to have the folder with your photos open. So here are my photos. And then I have my data sheet. And you may, your data sheet's probably going to be a paper copy. I'm just showing you this on my computer screen so that I can walk you through all of this. So once I have all those things, I'm logged into Bumblebee Watch. I'm going to go to this new button here, and I'm going to choose Add Sighting. You'll notice that there's two main projects that are pointed up here at the top. There's Bumblebee Atlas and there's Bumblebee Watch. Because the data that we're collecting for the long-term monitoring protocol is a little different than the Bumblebee Atlas that we've been doing in the past, we're going to go back to selecting the specific project. So you're going to use this regional projects dropdown, and you're going to choose PNW Bumblebee Atlas. When you do that, it'll open up this form, and it will have all of the same fields that match your data sheet over here. Okay, you're going to start by entering what we call the checklist information. So this is the information about your site. So the first time that you go to a site, you're going to choose add a new location, and you will name this site workshop demo, or whatever you called it on your data sheet. In the upper left hand, I called this workshop demo. So I'm going to write that here. And then I was good and I took my latitude and longitude while I was there. So I can just type those directly into the data sheet here. So I'm just copying what's in my data sheet into the data form. For longitude, remember that you always need to add a negative before the longitude. In the Western Hemisphere, longitude numbers are negative. The website will remind you that if you don't do that, but please try to remember to do that, because we do end up with sites in Mongolia from time to time when folks forget to do that. So I'm just, again, copying that data down. And then, so if you didn't happen to write down the latitude and longitude, you can click this select a map, and it will bring up the map, and you can interact with the map, you know, and find the location and just click on it, and that will drop the pin and automatically enter the latitude and longitude here. So that's also an option. Because we're going to be using the same site again and again and again, the next time you go back, you're not going to use this add a new location box. You're going to go back to an old location. So you'll choose from your locations, and in here, you would go to this drop down and look for your site that you called workshop demo. I haven't submitted this yet, so it's not actually in here, but when you return, you would choose workshop demo instead of actually selecting or entering a new site. Then you're going to choose the accuracy. So if you're sort of take that latitude and longitude at the middle of your hectare, remember that a hectare is about 100 meters by 100 meters. So if that was taken at the center of your plot, you could choose 25 to 50 meters. Maybe you'd go up to 50 to 100 if you needed to. Then you're going to add the date of your sighting. You can see on my data sheet that I did this in the future on June 1st, because that's the first day that survey season's open, but I'm just going to enter May 1st for the purposes of this demo. The survey type here, I'm going to choose point. Collection method here, I said that I captured bees that look different, so I'm going to enter that. My survey was 1 hectare. I had 1 surveyor. My survey started at 12:45, so I'm going to enter that in, add the PM. My survey ended at 1:45 PM. I'm going to enter that. My survey length was 45 minutes. Survey temperature was 63 degrees Fahrenheit. I sure hope it's warmer than that on June 1st. The cloud cover was 15%. The wind speed was 4 miles per hour, so I'll choose 0 to 5. If you had any weather notes, you could add them in here. And then I'm going to add the habitat type of the survey area. I said this was a grassland, so I'm going to choose grassland. And then the surrounding area, I'm going to choose grassland and then shrub scrub. And because I didn't choose one for tertiary, I'm just going to leave that one blank. How much of the survey area had flowering resources available? I said 60, so I'll choose 60. I saw bunch grasses and rodent holes and bare soil and leaf litter, and I will enter those here. And then for evidence of mowing, I did not see mowing. I did not see agriculture. I did not see insecticide. I did not see herbicide. I did not see evidence of fire. You'll note that right now, I cannot see anything here about honeybees or grazing. My website developer is still working on those fields. They will appear here just as they appear on your data sheet. So there will be an empty numerical box for your honeybees, and there will be the option to select these 4 choices for your livestock grazing. Then you're going to choose the number of flower species. On mine, I said I saw 3, so I'm going to choose 3 here, and it will open up the boxes that I'm going to enter. And so now I can move to the second page of my data sheet. And you don't need to fill in the common name of the plant, ideally just the scientific name. So here I'm going to put in Potentilla gracilis. And you'll notice that there is not currently an area also to enter the percentage of total plants. That box is also in development and will be there by June 1st. It'll actually be there by next Wednesday, but it's not currently ready for production. So again, here I'm going to put lupinus species, and then the third one is pedicularis. And then mine was not a volunteer effort because I work for Xerces, but if yours was, you would fill out those things. And then I'm just going to enter my name here and hit submit. And then it's going to bring me to the area where I'm going to start entering the bees that I saw. And you'll look on my website here, or I'm sorry, on my data sheet here, and you'll see that I caught 2 bees. And I've organized those already into a folder here. So I'm going to open up my folder of my window, and I'm going to choose the folder where I put the photos from this survey. And so I'm going to sort of drag this window box over here and put it underneath here so you can see it. And what I'm going to do for this first bee is I'm going to first just grab my photos. So I can hit select photos, and it can open up a new window, or you can just drag the photos over. In the past, we were limited to 3 photos. I think now we can take up to 5. So I'm going to take one that shows the profile, one that shows a close-up of the cheek, one that shows the back of the abdomen, and it goes up to 9 here. I'll choose that one as well. And then I can just drag these over to the website and drop it, and they'll automatically start updating. And when you see the blue bar disappear, it means that it's already uploaded. I know that this is Bombus occidentalis, so I can choose occidentalis. If you don't know, you can just choose SPA here, or bumblebee. So that's Bombus species. Or you can guess. It doesn't matter. We're going to look at the photos and correct them anyway. I know this is Bombus occidentalis, so I'm going to choose that one. You can also still use the identification tool. So if you want to use that as we have in the past, the different colors on the face and the thorax, you can help narrow it down that way, and use the identification tool if you want to, but you don't have to. This count and reference ID are not relevant for most of you, so you won't be using that. If you will be. And then for sex, this is new also. In the past, we weren't submitting this information, but now we are asking you to. So you're going to choose whether this was a worker or a queen or a male. In this case, I can see pollen on it there, so I'm going to say worker. And then the floral host here, I'm going to put Potentilla, Procellus. And then I'm going to, in the observation notes, I wrote, this is one of those ones where I had the first detection. So I'm going to write first detected at 1:11. And since I know the start time of your survey, I'll know that it took you, in this case, 21 minutes or something like that to find this bee, which will be helpful. And then I can click add sighting. And then I have my checklist. I have 1 bee that's added to my checklist. And if I have another one, which in this case I do, I can click add sighting and add a new one. So now I'm going to go and choose photos from my second bee, including all the features that I need to see. And I can drag those and drop them. I can choose the species. This is Bombus bifarius. This is also a worker. The floral host was lupinus. And if I had any observation notes, I could put them here. I don't have a first detection in this case. There's nothing that I necessarily need to say. So I can just leave it at this, and I can just click add sighting. And then you're going to continue in this way until you've done all of your bumblebees. And at that point, you will be done. You will have already entered all of the information from your checklist, and you will completely be finished. One of the things that I want to just show you is the other thing that you will see is when you're submitting your data, it's possible that you may do a survey and not collect or see any bumblebees. If you have done a survey and not seen any bumblebees, there is an option for you to say so, and I should have showed you that. I already have bumblebees in my survey, and I'm not going to refill out the form, but there's a checkbox for you to say, I conducted this survey and I didn't see any bumblebees. And so if you did that, we want you to submit all your data still, and then just make sure to check that checkbox that says, I saw no bumblebees. And then you should be able to see, if you go to your profile, when you're done, you should be able to see your recent bumblebee observations and your checklists. And so here's my workshop demo checklist here. So I can click on that and I can see all the information that I entered. I can see the location, I can see the bees and whether they've been verified or not, and interact with that. And so the other thing I'll just note is with the new Bumblebee Watch website, we also have updated the maps a lot. And if you want to like compare different species of bumblebees, you can do that on the map now. And it's anyway, it's just a very the website is a very cool upgrade. There's lots of new toys in it, lots of fun things to play with. So I would encourage you to, in addition to learning how to submit your data, I encourage you to play with the new Bumblebee Watch data set or webpage. There's a lot to see there. And I think that's it. That's a nice demo of how to participate in the project, or how to submit data from the project rather. And I think I'm going to pass it back over to you, Molly. Is that right?
I think you were going to carry on through because we have volunteer ID cards.
Got it.
After that.
Thanks for the reminder.
We do have one question that might be good to answer right now.
Yeah.
Someone asked if a single site is boggy at the time of the first survey and then dry grasses and forbs on the third and fourth, is there a way that they should note that when entering data?
Oh, you mean the habitat type?
Yeah.
I don't think bog is really an option. And if it's boggy but still dominated by grasses, I think you would still call that a grassland. Like it wouldn't be a riparian area, and I don't think wetland is an option. But if it is an option, I think it's okay for you to choose wetland in the beginning and change it to grassland later, as long as you're at the same location. I think we can parse that out at the end. But yeah, do the best you can to describe it at the time that you're doing your survey is what I would say. Yep, it's a good question for sure. This is just to point out that, you know, like this workshop is one resource that we've made available to you. We're recording it. You can watch it again and again and again if you want to see all of these details. Our website also has a ton of resources on it, including a page just directed to resources where you will be able to find this webinar, where you can find equipment suggestions, where you can find the data sheets. You can learn how to ID bumblebees, look at the field guide, learn how to take photos. And permitting isn't really a huge issue in the Pacific Northwest, but some of our other projects it is. So lots of resources available online. We also have a volunteer card that is available to you. These are lanyards that can help you get recognized out in the field while you're doing surveys. You know, in case someone comes by and asks you questions, you can show them your ID card. There's a phone number on the back. You can have them call if they have questions about our program. And if you scan this QR code or type in the URL at the bottom of the screen here, you'll be taken to a form where you can fill it out. And if you fill that out by May 19th, we will get those in the mail to you, and they should arrive by around June 1st. So if you'd like a volunteer card, these are optional. You don't need to have one. Some people like to have them. Other people couldn't care less. So it's completely up to you. But if you're interested, please go ahead and request those. I'm going to zoom past this now. If you need the URL or want us to share it with you, just ask in the chat or send us an email, and we can send this right back out to you. The participant handbook is also brand new. It's available, that it describes the new protocols. It's also available on the website. It's available on the PNW page, and also on the print resources page. It's in a few different places. And the data sheet is also there. I'm just going to quickly go back and just show you the print materials web page here. At the top, you see that there's a data sheets, and most of the Bumblebee Atlas projects, like in the other 17 states, are going to use this data sheet on the far left here. This is the data sheet that you want to use that'll have that information about honeybees and the different plant stuff and the grazing. So you want to choose this long-term monitoring data sheet over here on the right, or if you scroll down to the Pacific Northwest print materials, here's the handbook, and here's the long-term monitoring data sheet and some other resources that are available to you. And the other place that you can find these is if you just go to PNW here and go to our regional page, which you'd find by going to Get Involved, Find Your Atlas, and then choosing Pacific Northwest. If you scroll down to the bottom, here is the handbook, and here is the data sheet as well. And then also there's if you want help, you know, in the past we've shared the Google map that has the grid cells and the long-term monitoring sites. If you want to have this for yourself to edit and look for sites, you're welcome to download that or use this as a resource as well. Lots of folks have used that in the past, and it's still available for you. And then the contact information for Molly and I is on the bottom of this page as well. So I think maybe am I passing it back over to you now, Molly? Or yeah,
I think all that's left is questions.
What's that?
Oh, I said, I think all that's left is Q&A time.
Oh, great.
And I've been answering some in the chat. There are a few I wanted to highlight because I thought that it might be helpful for multiple people.
Yeah, great.
One is that if you're counting honeybees, but you don't find any, should you write that in there? And the answer is yes. It'd be helpful to put a 0 if you don't find any, so we know that you didn't just skip counting.
Yes.
So 0 in there is definitely helpful. Let me see. And also with grazing, if you don't find any of those signs, you don't have to write none. You can just not circle any of the characteristics like hoof prints and cow pies and things.
Yeah, and one of the options when you go to submit that will be none of the above. So if you didn't see any of those features, you would just write none. You would just choose the none of the above option.
And then another good question that might be relevant to multiple people is if you can't identify a plant while you're in the field, it's a good practice. You can take a picture and note the number of that picture and then identify it once you get home. So someone asked about that, and that's absolutely something you can do.
Yeah, and something I also wanted to mention when I was entering my data and showing you that process was that you can, you know, submit up to 5 photos, but you know, oftentimes you can get all the features we need to see for ID in 3 or 4 photos. And if you want to add the photo of your plant in there, that's pretty good practice as well. It's certainly not mandatory, but if you have an extra spot to throw a photo of the plant in there, it helps us. Sometimes we just randomly go through and look and make sure people are identifying their plants correctly. And so that's helpful. Again, it's not a necessary thing to do, but it's another way to get another pair of eyes on your photos, on your plant photos.
There's one other question I just saw that came up. Somebody asked, they say, I recall I previously was not able to access the participant handbook, but would love one. And I don't know if that's a question about getting a print copy or accessing it online. Rich, do we plan to print any this year to share with people, or is it going to be mainly an online resource?
If you would like a hard copy of the handbook, send me an email. We can print them out and ship them out to you. I can have Karina do that in the office. So if that's something that you would like, we can make it available to you. Just send me an email. But I'm just checking right now, the links are working. So the download, if they're looking for the PDF, it's definitely working. And literally, like, I put it up an hour and a half ago. So if you were looking for it more than that ago, you wouldn't have found it, but it's there now. Or I guess more than an hour and a half before the webinar. We just
not half an hour in.
Not during the webinar. I didn't.
Good to multitask.
Yeah.
Somebody else asked if multiple people people can adopt one cell, and yes, they can. And that's great to have a backup. Plus, you're really unlikely to end up surveying the exact same spot. So having data from 2 different locations within a larger cell is a good thing.